This video is about the second session of hearing series, the auditory central nervous system. In the last session, we talked about how the inner ear transforms the sound into an electrical signal. Here, we will start from after the cochlea. The acoustic information is processed by neural pathways. The neural pathways extend from the ear to medulla, the pons and the midbrain of the brainstem, and the thalamus to the auditory cortex. In the auditory central nervous system session, we will focus on the sound information, the cochlear nuclei, superior olivary complex, inferior and superior colliculus, auditory cortex, and sound recognition. This video will cover the sound information to superior olivary complex. First, let's talk about what kinds of information sound has and what is the role of this information. In many species, hearing serves as a basis for communication. Not only for the communication, but also it helps to alert to the presence of unseen dangers or opportunities. Additionally, by the means of the alert, hearing is crucial for localizing and identifying sounds. Having two ears is a great advantage for localizing and identifying sounds. Ears can extract the features of the sound, which are timing and intensity. A sound source at different positions affect the two ears differently. Sound arrives earlier and is more intense at the ear near the source. Also, the size of the head determines how the time delay and intensity difference between two ears. Therefore, ears use these kind of features to extract the information about where and what of the sound. The sounds in different positions arrive at different time on both ears. Therefore, the interaural time difference is related to the location of the sound source in the horizontal plane. After projecting the sound information into a neuronal signal, the information is conveyed by the cochlear nerves. The cochlear nerves contains two groups of fibers. 95 of myelinate fibers that receive input from inner hair cells, and 5% of unmyelinate fibers that receive input from outer hair cells. Myelin is a lipid-rich substance that surrounds nerve cell axons. It decreases capacitance and increases electrical resistance across the axonal membrane of neurons. Let's bring some simple example. Think about you're holding a hose with water coming through. What will you do if you want to make the water come out faster? You need to press a hose. By pressing hose, you increase the resistance of water and decreases the amount of water going through. However, the speed of water is definitely faster. Now, we can understand better that myelin increases the speed of electrical impulse propagation along the axon. Therefore, each myelinate fiber detects a narrow range of frequency. By the way, unmyelinate fibers integrate information from a relatively wide region of the cochlea. The cochlear nuclei can be divided into ventral and dorsal cochlear nuclei, and of course, their role is different. Here, we need to talk about the functions and pathways of three neurons in the ventral cochlear nuclei. Their name is octopus cell, Busch cell, and satellite cell. Octopus and Busch cells are able to respond with rapid and precisely timed synaptic potentials. Octopus cells detect coincidence firing which comes from periodic sound such as vowels and musical sound or broadband sounds. Compared to octave cell, Busch cells encode the temporal fine structure of the sound. These are the information about the relative time of arrival of input to the 2 ears, and this information is used at the next step synaptic stage to form a map of the time differences between 2 ears. They project information to the superior olivary complex. In contrast, stellate cells track modulation in intensity that is known to be important for understanding speech. They convey the information to the contralateral inferior colliculus. The cochlear nerve fibers also extend into the dorsal cochlear nucleus. The dorsal cochlear nucleus help interpret spectral cues for the localization of the sound source. To make full use of spectral cues, differentiation with information of head and ears position and the sound source information is needed. This information called predictable and unpredictable cues. The predictable cues come from body movement. The somatosensory and vestibular information about the head and the ear position descending from higher levels of the nervous system. Therefore, this information can serve to identify the predictable cues. This cues is handled by molecular layer of the dorsal cochlear nuclei. Unpredictable cues are that more interesting and informing about the location of an external sound source. This cue is handled by the deep layer of dorsal cochlear nuclei. The process of both molecular layer and deep layer are combined in the neuron of the dorsal cochlear nucleus, the fusiform cells. The results of these processes from the cochlear nuclei convey to the superior olivary complex. To localize the sound source, neurons in the superior olivary complex compare the activity of the cells in the cochlear nuclei. Separate auditory circuits of superior olivary complex detect interaural time and intensity difference. Medial superior olive circuits are responsible for interaural time difference, and lateral superior olive circuits are responsible for the interaural level difference. The cochlea cannot capture the arrival time difference of the sound, but in the medial superior olive creates a map of the interaural base by a comparison of the timing of firing in responses to the sound from the 2 ears. Here comes the important interaural time difference model, Jeffreys model. There are an array of detectors getting input from 2 ears. The inputs are transmitted through the delay line with systematically differing lengths. When the sound comes from the front, the input follows through the delay lines and stimulates all the detectors. Eventually, the middle detectors has the highest activity because they got the input from both delay lines at the same time. However, when the sound comes from the left side, the right side input follows the line with a delay compared to left one. This is Jeffreys' model and how the medial superior olive maps the location of the sound source. The difference between this spatial map and those in other sensory systems is that this map is not the result of spatial arrangement of input like retinotopic or somatosensory map, but is inferred by the brain from computation. When your ears receive the sound, the ears will face the intensity difference effect, which is the head shadowing. The head shadowing is literally the effect of the sound wave are diffracted by head. This means that the intensity of the sound is lower than that is not diffracted. So, this effect causes interaural level difference. This difference is detected by the medial nucleus of the trapezoid body and the lateral superior olive. The lateral superior olive performs the first of several integrative steps that use interaural level difference to localize sound. Differences in the location of the sound source generate differences in the firing rate of the neurons. The balance between excitation and inhibition is reflected in the firing rates of the neurons in the lateral superior olive. You need to consider balancing. To balance excitation and inhibition, stimulate by the same sound. The ipsilateral excitation and contralateral inhibition must arrive at the lateral superior olive at the same time. The role of the efferent fibers increase the representation of signal and noise. They reduce the sensitivity of the cochlea and project it from damage by the loud sounds. Olivocochlear neurons form a feedback loop from the superior olivary complex to hair cells in the cochlea. Thank you for watching. That's all for the first part of auditory central nerve system. The next will be the second part of auditory central nerve system. See you then.